Dr. Brandi Kamermans Yintia, Nahobani Nishlin. Hello, my name is Dr. Brandi Kamermans, and my clan is where the gray streak ends. I am a senior researcher at the Salish Sea Research Center, which is located on Northwest Indian College's campus in Bellingham, Washington. I have been working alongside Justice in the last year and helping her learn about environmental DNA and how it can be used in a good way for natural resource management for tribal communities. In the last year, Justice has helped collect DNA from marine and freshwater environments. She has learned how to extract DNA using chemical processes and commercialized kits. She has also learned how to do polymerase chain reaction experiments and quantitative polymerase chain reaction experiments. The work that Justice has done in the last year will leave a lasting archive of the environmental DNA that was present in these environments for a very long time. These are very precious samples, and they leave behind something for the future if Lummi Nation were ever interested in species that were either invasive or endangered in these environments in the future. So currently, Lummi Nation is interested in longfin smelt, and they are interested in European green crab. One is an endangered species, and one is an invasive species. But the filters that Justice collected not only contain potentially the DNA of these organisms, but any organism that was in that aqueous environment at the time that we collected them. And as a Native American scientist, I find it very important that community members know that environmental DNA and molecular techniques, including genetics, are a powerful tool that can be utilized for natural resource management. And I hope that students like Justice become interested in genetics as a tool, because we are at the cusp of a lot of breakthroughs with environmental DNA and natural resources management, and I want tribal communities to harness both data sovereignty and governance over samples that are collected on tribal lands, and data that can inform better management practices at a tribal and a federal level. So it's been a wonderful experience working with Justice in the last year. She's learned a lot about environmental DNA and how to track both invasive and endangered species using environmental DNA. And I hope that with this project and future projects that we can empower tribal communities, and we can do research in a good way on tribal lands. And thank you very much.
Now CM Sweet Senahila Juana Senatachula Tia Kaya CM and Asachula. Hello, my name is Justice Black Williams. My indigenous name is Tlatli Palis. I am an enrolled member of the Lummi tribe, which is located in Bellingham, Washington. And I'm currently getting my BS in Native Environmental Science at Northwest Indian College. And I've also been a student intern at the Salish Sea Research Center since June of 2022, which is an independent lab at Northwest Indian College that has consistently been teaching me different research techniques, methods, and protocols. When I first started this internship, I was completely new to this field of study. I didn't even know how to use a pipetter, let alone complete a protocol. But with all that in mind, I was welcomed with open arms, and in that time, I found a passion. I started off by collecting field samples at our marine sites using a Smith-Root eDNA backpack sampler in search for longfin smelt, or better known as hooligans. Once the filters were extracted from the backpack, I used the Kaijin DNAZ blood and tissue kit, modified a step with the Kaya shredder kit, as well as eluting 2 times 50 microliters of low E buffer to collect the environmental DNA. eDNA is a non-invasive tool we use at SSRC to detect species in our environment. You can collect eDNA from samples of the soil, air, or water. And that sample can carry a genetic code of a variety of species, which can tell us a lot of information about the ecosystem in that area. Next, I learned how to collect genomic DNA. Genomic DNA is different from environmental DNA because it is extracted directly from the specimen. In this case, we extracted gDNA from longfin smelt livers, and we chose the liver just because it is packed with DNA. In this protocol in particular, we use the Beckman Coulter DNA Advanced Kit with a modified step of using DTT with protein SK and lysis buffer for the master mix. So once I learned the preferred eDNA and gDNA methods, I then moved on to learn how to extract DNA using phenol chloroform isomal alcohol protocol to detect the invasive European green crab in a man-made seawater pond on the Lummi Reservation, which has just been a huge problem for us, for our community. Once I'm confident doing a standard operating procedure, a SOP, I move on to a new one. Simple as that. Taking all that new information and that new knowledge I obtained with me. And I love what I do here. I really do. This internship is what drove me to do a program of study change request into the Native Environmental Science program at Northwest Indian College. And although I have just begun this program of study, I am continuously putting the information I attain into action here at SSRC and taking the information I gain at SSRC into the classroom. All of this made me learn I want to be a part of something larger than myself. And that's what I feel like I'm doing here. I aspire to help create and maintain a sustainable way of life like my ancestors before me. As someone who is indigenous to this land, I have a responsibility and an entrenched respect for the natural world. And through this research, I feel as if I'm personally putting in the work to help innovate environmental solutions for my community.
Welcome to Northwest Indian College. This is where I'm currently getting my BS-NES degree. And this is SSRC. Let's go. So today we're going to be doing a PCR protocol, and I'm just going to input the data before we start. Now we're going to do the protocol. I really sped this along because it is a whole day process, so you just get the gist of it. Yeah, we're getting all of our samples ready to test to see if we can get any hits of European green crab in the Red River at Lummi Nation. And here we're putting it in the PCR machine, and we already have a template ready. We just click on the EGC method and get it started. It'll be done in 2 hours hour and 15 minutes. And then we're going to make the gel. So we start out by putting 4 grams of agarose and then dumping that into a beaker, and then putting SB buffer, 100 milliliters in there, and then also Millicue water, another 100. Then we're going to put it in the microwave for 3 minutes, checking every 15 seconds. And we do not want it to boil over. And right now is just getting all the bubbles out and then using to get this last ingredient in there. It's really toxic. Then we're going to pour it into these mini horizontal units for the electrophoresis systems. They're by Fisher Biotech. Then we're going to get all the bubbles out using this plastic ladder well maker. And then this is the whole reason of being why we're doing this protocol in the first place, and really tells you the gist of it. So pause to read. Now we're taking out the plastic well maker, and then we turned it around so it's going to be running to red. And then we use, what do we use? A low molecular weight ladder and a 50 base pair ladder at the 2 end wells. And then we're